Hello, listeners. Yamina here with our very first Dr. GPCR podcast episode of 2021. I know it's already February, but please allow me to wish you all a happy new year. I usually thank the guests in our team at the end of each podcast episode, but today I'd like to start with a few thank yous. First of all, thank you to all of you listening, reading, and following Dr. GPCR on social media. Thank you to our team for being so dedicated, namely Attila Forrest, my partner in crime, Shivani Sajdev, and Jin Chong, our science communication experts. I would also like to take a minute to thank Dr. Randy Hall for his support. And last but not least, thank you to Dr. Bob Lefkowitz for sharing his stories and his time with me. Before we open Season 2 of the Dr. GPCR podcast with this very special episode, I wanted to let you know that the Dr. GPCR February newsletter will be in your inbox soon. If you haven't subscribed yet, visit drgpcr.com/newsletter. Also, be on the lookout as we will be launching our brand new monthly Dr. GPCR virtual cafe starting in March. You'll be able to join us live for 1 hour of GPCR talks. If you'd like to present your work or your company's technology during a GPCR virtual cafe, email us at hello@drgpcr.com. More on the topic coming soon. And now, let's dive into this very special episode with Dr. Bob Lefkowitz. Hello, everyone. This is Yamina from the Dr. GBCR podcast. I am ecstatic today because I have with me see, I'm already stammering, the one and only Dr. Bob Lefkowitz from Duke University. I don't think Bob needs an introduction. Everyone in the field knows who he is, so I'm just going to jump in. Hi, Bob. How are you?
Delighted to be with you today.
Thank you so much for being here today. I was so excited when I learned that you had time in your schedule to come on board and to talk to us. So let's start at the beginning. I got an email telling me about the book that you're going to release soon. Tell us a little bit about the book, and then we can jump into the questions I have.
Okay, that sounds wonderful. So may I share a screen?
Absolutely. Absolutely.
Okay. Host disabled participant screen share.
Well, let's jump into the questions, and I'll enable it in the meantime. So everyone,
say a word or two about the book.
Of course.
Showing the picture, and then we can show the picture later. So this is a book called "A Funny Thing Happened on the Way to Stockholm." It's going to be published on February 2nd by Pegasus Books, and it's available for pre-order now on Amazon. And it's basically written together with Randy Hall, who was a postdoc of mine in the 90s. He's now a professor of pharmacology. I'm sure many of you are familiar with him. Anyway, we tried to, the inspiration and the model for the book was a book probably known to many of the people in the audience called Surely You're Joking, Mr. Feynman, which is a book that Feynman, the Nobel Prize winning physicist, wrote together with one of his former acolytes. So basically, it's sort of the story of my life and career with the science woven through as well. And it is basically a story of how I came to be a scientist in the first place, which was, we can get into later, which is quite accidental since I was planning to be a physician. So I think it will be a particular interest to this audience because, as I say, the whole scientific evolution of the field is in there, but that's just the backdrop. Mainly it's about sort of me and my career and who I really am, as opposed to the image that people may have.
That's exactly what we want to find out today. So we're gonna dig into a little bit deeper into who you really are. Can I ask, when did you first get interested into science?
Well, I first got interested in medicine, actually, not science. I would say when I was by the time I was 8 years old, I had basically decided that my career path would be that of a physician. I would say that the word calling, we associate the word to have a calling often with clergy, but you can have a calling to almost anything. And in my case, by age 8, inspired by my family physician, Dr. Joseph Feibush in the Bronx, who made house calls, I had decided that I wanted to be a physician. There was no doubt about it. And I never wavered from that. In subsequent years, I realized that by the time I was in junior high school, I realized that the foundation of medicine was science. And so I became obsessively interested in biology. I had a little toy microscope, which actually worked, and I would tear the wings off flies and look at that, or samples of earth looking for microorganisms. I had a little chemistry set where you could mix things together. So I was interested in science early on, but not in research. I loved knowing the science, but I had no particular interest in being a scientist, nor in fact did I ever do any research in school, not in high school, not in college, not in medical school.
It's so interesting. Did you have any biology professors that inspired you to dig in more into the knowledge and kind of understand what surrounds us?
Yeah, absolutely. As with most students, there are key professors that along the way that really inspire them. So in college, there was a professor, a very, very young assistant professor who had just joined the faculty in chemistry at Columbia, where I was, by the way, a chemistry major, named Ronald Breslow. He passed away within the last few years, but was one of the most distinguished went on to a career as one of the most distinguished academic chemists. And he taught an advanced seminar in biochemistry, which I took. And I really fell in love with the subject matter. And then in medical school, the guy who really inspired me, who also passed away recently, was Paul Marks, a name that may be known to many, who was for many years the head of the Memorial Sloan Kettering. He was a physician scientist and had been very involved in working out the biochemistry of some of the hemoglobinopathies. He taught the introductory course in clinical pathology. But rather than just teaching us the rote stuff, he would actually assign readings from the primary literature, which many of my classmates hated. They just wanted the facts, but I just found them absolutely fascinating, as did my classmate, Harold Varmus, who was a good friend. You may have heard of him. And Harold and I both idolized Paul Marks. So those two professors, Breslow and Marks, really inspired my interest in biochemistry.
So you mentioned that you got really, as an 8-year-old, you wanted to be a physician based on the family physician who made house calls. Then you got interested in science and research and in biochemistry. So you're in med school. What takes you over from a medical degree to research?
So I wrote an autobiographical essay a few years ago, which some of your audience may find interesting. It was published as sort of the front matter in the annual review of pharmacology a few years back. And it was called A Serendipitous Scientist, because it was sheer serendipity that diverted my career from being a full-time academic physician to becoming a physician scientist. So I graduated in medical school in 1966. This was at the height of the Vietnam War, when there was a doctor draft. Now, there was a lottery draft for all men over 18, which meant just that. It was a lottery. Depending on what your number was, you either went in or you didn't have to go in. But for physicians, there was no lottery. The doctor draft mandated that all physicians, upon graduating from medical school, would get a further 1 or 2 year deferment to get an internship and maybe a year or two of residency training. And then everybody went in. The only question was, what branch of the service did you go into? It was a 2 year commitment, and you spent 1 year in Vietnam. Now, the war, as many of you may have read about, since I assume a lot of the audience wasn't, observing it firsthand, the war was extraordinarily unpopular. It was viewed by many of us as immoral, even illegal. There were demonstrations, riots in the street. So most folks of my ilk didn't want to, support that war. But there were very, very few legal ways to avoid military service in Vietnam if you were a recently minted physician. One of the very few was to gain a commission in the United States Public Health Service. The United States Public Health Service was at the time considered one of the military branches. And so if you served there, it counted as your draft service. Of course, why would that be desirable? Well, because many of the assignments that the Public Health Service had were within the United States. And so, for example, prison physicians, service at the NIH, taking care of the patients at the clinical center who were in for studies, service at CDC, which everybody knows about these days, and other such assignments. So you can imagine how extraordinarily competitive it was to get these commissions. Everybody wanted them. And so they basically got the best and the brightest. Fortunately, I had very high academic standing and good recommendations, and I came from Columbia, which was, a terrific school. And so I did get the commission. And so in 1968, after 2 years of house staff training, I was drafted into the Public Health Service as a lieutenant commander and assigned to the NIH, where I spent about 20% of my time taking care of patients. And 80% of my time I was assigned to a laboratory with two mentors, Jesse Roth and Ira Paston, where I had my first experiences in research. Things did not go well. For the first year and a half or so of the 2 years I was there, nothing I did worked. And this was a very disturbing experience for me because, to be frank, I had never really experienced any sort of sustained failure in my life to that point. Things had generally worked for me. And now, despite my very best efforts, I was making no headway on my project. But finally, after about 18 months, I had a breakthrough, and then things started to work. And I got a taste of just how, kind of fun and exciting it can be to be doing research. But I was not fully convinced, put it that way. And so after 2 years of service, I went off to complete my medical training, now in Boston at the Massachusetts General Hospital, where I spent 3 years further training in internal medicine and cardiovascular disease. But it was during the first 6 months of that I had my epiphany. During the first 6 months back in clinical medicine, I threw myself into what was an extraordinarily hectic and busy clinical life. I loved it. I had always loved clinical medicine. And, to be honest with you, I was pretty good at it. But I realized during that time that something was missing. Mm-hmm. And I finally came to the realization that what I was missing was the day-to-day excitement and stimulation of experiments, having data, and wrestling with a research problem. And so I found myself a mentor after 6 months. And for the remainder of my time at the Massachusetts General Hospital, I spent, I basically moonlighted in the laboratory while doing my assigned clinical rotations. Now, and then I went to Duke in 1973 to begin my independent career. I just want to point something out about my time at the NIH. The group of us who served at the NIH in the Public Health Service came to be known, and we use this sort of sarcastic moniker ourselves, as the Yellow Berets.
That was my next question because I knew something about it, so please.
Right, so the Yellow Berets, what does that mean? Well, it was a takeoff on the Green Berets, of course, who were the elite commando forces that we had, special forces. Yellow, of course, is taken from the classic color of cowardice. Of course, most of us felt we were there not because we were afraid to serve in Vietnam, but because we thought it was morally wrong to support the war. So we were known as the Yellow Berets. The impact of that program if you just take from 1964 to 1972, an 8-year period. During that time, 10 of us, all young physicians, none having any significant research experience before going there for a couple of years, 10 of us would go on to win the Nobel Prize.
Wow.
In my class alone, the group who was there from 68 to 70, so how many could that have been? Maybe 100 people. That group contained, in addition to myself, Michael Brown, Joseph Goldstein, Harold Varmus, and oh, by the way, Tony Fauci.
Of course.
Okay, and we were all there from 68 to 72, and we all left. I'm sorry, 68 to 70, and then we all left. Tony was the only one who came back, and he came back a few years later. So that was a remarkable experience, and it was that serendipitous event of the Vietnam War and going to the NIH when I did, not because I was dying to do research, but just because I didn't want to be dying in Vietnam.
Of course.
It was that serendipitous event that basically changed the entire course of my life and career.
That is so fascinating. And I've said this to many people in the audience. So I worked at NIH for about 3 years, and I actually was in a smaller group that actually belongs to Dr. Fauci. And for a long time, I don't think people knew who Dr. Fauci is, but now everyone around the world knows who Dr. Fauci is.
You knew Fauci before he was Fauci.
Something like that. I've seen him. We've been having these monthly lab meetings that were at 7:15 in the morning because Dr. Fauci was very busy all the time. But yeah, he's just a phenomenal person.
He was always famous for his work ethic.
He is.
To this day, which we continue to see on very obvious display every day in all kinds of media. Tony and I have been good friends for the better part of 50 years. I have been in communication with him on half a dozen occasions since the pandemic started. I have several Tony Fauci stories in the book, including at least one photograph of me and Tony together from probably 25 years ago.
Fantastic. And I think it's just the book comes out at the right time, but having Dr. Fauci's stories in your book, I think will also be of big interest to the readers. So what I wanted to ask you is, so you're at NIH, you get into doing research in the lab, and for 18 months it doesn't work. What were you working on?
Okay, so again, a wonderful piece of serendipity for me. So I arrived there, and it's hard for people today to imagine what the field of signal transduction was like in 1968. The big guiding principle in the field was the discovery, having been made maybe a decade before, or not even less, by Earl Sutherland, of the enzyme adenylate cyclase and its product, cyclic AMP, which seemed to mediate the effects of adrenaline and glucagon. And at the time, that's all we knew about how it worked. At the time, what many people were working on was trying to find other hormones that stimulated adenylate cyclase, and they were finding them. And that was the big thing that people were doing. Sutherland, the idea that there might be specific receptors for hormones was floating around, but was not a popular idea. And in fact, Sutherland published on this. Sutherland's idea was that whatever a receptor for a hormone might be, it was some sort of allosteric binding site on the enzyme adenylate cyclase. Okay? Now, my mentors, Jesse Roth and Ira Paston, both of whom you may have heard of and who both went on to fabulous careers. They were both fairly young at the time. They thought it might be possible to get at, Sutherland's idea about, the fact that there might be binding sites on the enzyme by radioactively labeling a hormone and studying its binding to preparations that contain the enzyme adenylate cyclase. And for other reasons, they had, in their research groups, they had been passaging an adrenal cortical carcinoma in nude mice. And so the task I was assigned was to see if I could develop such a radioactively labeled binding assay of a hormone to a membrane preparation from these tumors. Now, it was an adrenal cortical tumor, and it was ACTH sensitive, adrenal corticotropic hormone sensitive. So the idea was to radioactively label ACTH with 125I, separate the monoiodo radioactive ACTH from the non-labeled ACTH, show that the radioactive stuff was still biologically active, and then do binding studies. And where I got hung up was separating the iodo ACTH from ACTH. So here you have a molecule of several thousand Daltons, and you have a one atom difference, right? I had monoiodo ACTH, and they wanted me to separate it from ACTH so I could bioactivity of my radioligand. And that took me over a year. I learned a lot about separation chemistry, as you can imagine, but it was brutal. And I did not deal well with the protracted failure. It was just absolutely miserable experience. But I learned a lot about how to deal with failure in research, because as anybody who's had a career in research knows, failure is our constant companion. And in fact, I give a little tutorial. I have a lecture that I give every couple of years at Duke to research fellows called How to Deal with Failure and Rejection in Science. So I did learn a lot about that. Anyway, so that was my introduction to the idea that there might be receptors and that development of radioligands might be an approach to trying to see if there really were discrete molecular entities which served as hormone receptors. So that's where that all began.
And did this experience influence your choice of research, or the choice of research topics that you went on pursuing at Duke?
100%. So I decided that studying, so I was convinced by my research, even though the proof was not there, but by the time I left the NIH, I was convinced that there was such a thing as a hormone or a drug receptor. And I don't know, it just seemed to me that maybe it was different than the enzyme itself. I mean, I had no evidence that that was the case.
Of course.
So when I made the decision up at the Mass General that, I really want to do research, I said to myself, I can't think of a better topic than receptors. We know nothing about them. We don't even know if they exist. But if they do exist, they're obviously the gateway to the cell. And boy, that would be a big impact if I could make headway with that problem. So then the issue became for me, well, okay, what receptor are you going to study? And I thought about that long and hard. I decided I wanted to study a receptor which was in some way related to the adenylate cyclase, okay? Because I would have an easy to assay, very proximate signal downstream of whatever receptor I wanted to study.
Yep.
But I was also a young, aspiring cardiovascular physician, in training. So I wanted something related to the cardiovascular system, which certainly ACTH was not. So I started thinking about what I might study. I thought about glucagon receptors because it was known that there was glucagon-sensitive adenylate cyclase in the heart. I even did a couple of experiments with that. But I realized, one of the things that really limited me at the NIH when I was working on ACTH was that there were no analogs or to study. In other words, there was just ACTH. So if I, for example, wanted to see if a series of ACTH analogs, for example, could displace my binding assay, in parallel with their ability to activate adenylate cyclase. I would have to literally make those analogs myself, for example, by limited proteolysis of the ACTH, et cetera. And that's what I did. And I came up with all of 3 or 4 analogs. But as I thought about it, and the same problem limited glucagon, but when I thought about it, the adrenergic receptor system had literally dozens and dozens of analogs that people had studied over the years. Yeah. And in fact, one of the interesting ironies in the field that I'm sure people don't realize is that by the time I was doing this work in the early 70s, even in the late 60s, people talked about alpha and beta adrenergic receptors. Raymond Ahlquist, a classical pharmacologist of mid-20th century, had come up with the idea of distinct alpha and beta receptors based on classical pharmacological experiments looking at orders of potencies of agonists, okay? And so, well, if it's this pattern, I'm calling it beta. If it's this pattern, I'm calling it alpha. And he used the terms alpha and beta receptors. But he didn't believe in receptors as anything other than a concept. And in fact, Ahlquist and I were at a symposium in 1973, before I had made any real headway with my research. And I was asked to present on what I wanted to do. And I talked about how I wanted to prove that there were adrenergic receptors and isolate them and find out what they are. And Ahlquist, who had won the Lasker Prize for his work in 1948, was my discussant. And he got up and he said, it's all very nice. He says, but he says, this is a figment of Dr. Lefkowitz's imagination. There's no such thing as a receptor. And he published this. You can look it up. And I cite it in my book. He said, there's really no such thing as a receptor. A receptor, he said, is just a pattern of forces on the cell membrane, which somehow, is able to figure out what's going on.
Magically.
And I remember thinking to myself as a young buck at the time, I'm going to show this guy, this son of a gun, and I'm going to show him. Well, I did, but it took a hell of a long time. So I decided, well, look, adrenergic receptors, in particular beta adrenergic receptors, what could be more germane to cardiovascular medicine? And there are all these analogs and agonists and antagonists available. Yeah. And I'm clearly going to need to do some chemistry here if I'm going to be making radioligands, and if I want to make photoaffinity probes. If I want to isolate the receptors, I'm going to need to make affinity chromatography resins. I'm going to need all sorts of, analogs. It's got it all. It's got the clinical relevance for a cardiovascular physician. It's got the basic tools of having all these analogs. That's the system. 50 years later, I look back on it, probably the smartest decision I ever made in research.
That's phenomenal. So you come up with the idea of going down this path. You're at Duke, and how did you start working on this? What was the first experiment that you did?
Okay, so while I was at the Mass General, I was trying to develop a radioligand to study the beta adrenergic receptors, and I basically wound up going down some blind alleys. And so by the time I moved to Duke in 1973, I really hadn't made any substantive progress on that. So I spent the first year with my initial team, and you want to talk about crazy stuff. So let me tell you how I built my laboratory. I came to Duke by myself, nobody, okay? My very first hire was Lee Limbird, who some of you may have heard of, went on to a distinguished career, provost, chair of pharmacology at Vanderbilt, provost at Vanderbilt. There's a Lee Limbird chair there. So she was my first postdoc. Second postdoc, Mark Carone. Okay, I think a few of you have heard of Mark Carone, James B. Duke professor. Third person to join my lab on day one was a young MD PhD student looking for a mentor, a guy named Rusty Williams, went on to discover the PDGF receptor, be elected to the National Academy of Sciences. And I could go on from there, but I won't. So that was my team. So I basically took on initially the responsibility for trying to develop a radioligand, because that seemed very, very risky to me. And I had the others working on a variety of what in retrospect weren't great projects. But I found the breakthrough finally came in the spring. I came in July 1, 73. The breakthrough came in the spring of 74 when I developed a radioligand called tritium labeled dihydroalprenolol, which 50 years later is still in use and is one of the common ligands that people use. And that was the first big breakthrough that really enabled everything else.
So how did you, or what did you base your hiring decision on? Because everyone knows, before you answer, everyone knows all of these famous scientists who came out of your lab. It's like a dynasty of Bob Lefkowitz's lab. What did you base your decisions on?
It's true. And here we come to an answer that I think will surprise a lot of people. Fate. Okay, why do I say that? I never met Lee Limburg before she came to my lab. The deal I negotiated at Duke included the fact that the people who hired me would pay for one technician and one postdoc for 3 years, or until I got funding. Okay.
Wow.
So I said to the guy who had hired me, who was the chief of the cardiology division, well, nobody's applying to me for postdocs. I mean, you know, but I said, can you look around? So he called me up. He says, you know, there's this young lady I interviewed, and her husband is staying at Duke for an orthopedic residency. She just got her degree in biochemistry from UNC. I think she'd be good. If you want, I'll hire her for you. I said, sure, that sounds good to me. That's how I came to Lee Limburg.
Wow.
Now, okay, Mark Carone. So sometime in 1972, I was publishing, nothing in retrospect that was all that exciting. I was publishing, and in 1972, I get a letter from a young PhD student at the University of Miami, Mark Carone. He says, I've read some of your papers. They seem interesting to me. And I was wondering, he says, I'll be finishing my degree in the spring. I'm French-Canadian, and I can get a very nice 3-year postdoctoral fellowship, I think, from them. Would you be interested in taking me on? Obviously, Carone was under the mistaken impression that I was a professor and not a postdoc myself. Well, you can be sure that I did nothing to disabuse him of this misconception. So I wrote back and said, sure, I'd be delighted to take you on. So at this point, I don't know what I was thinking because I didn't have a job myself the following year, but I knew I would get one because they were telling me they wanted me to stay at Harvard. Anyway, so he got the fellowship and told me. And then once I made the decision to come to Duke, I wrote to him and I said, good news. I said, you won't have to travel all the way to Boston. You only have to go half as far because I'm moving to Durham. And so that's how I came to Mark Carone. The third guy, Rusty Williams, walked in, and at that point, he was an MD PhD. He walked in right on July 1. He said, I'm looking for a place to do my thesis work in biochemistry. He said, I haven't found anybody who sounded all that exciting to me. Can you tell me what you're going to be working on? Okay. So I spun my yarn and told him my aspirations. He was interested. I knew virtually nothing about him, nor did I care. He was a warm body. So I said, sure, why don't you come on board? So that's how I got the first 3. Now, I'd like to think I had something to do with how they developed. Okay, but the other guy I love to talk about, who I never met before he came to my lab, was a guy you may have heard of. His name is Brian Kobilka. So Kobilka was a medical resident at Wash U in St. Louis, and he wanted to. He was thinking he wanted to be either a cardiologist or an emergency physician. So he was looking at cardiology programs, and he knew of my work. This was now a decade later in the 80s. And he saw I was publishing on adrenaline and adrenergic receptors, and he figured, well, adrenergic receptors, that has to do that would be of interest to somebody who's going to be in either cardiology or whatever. So he applied to the cardiology training program at Duke with the specific interest in working in my lab. So when he came for the interviews, I was out of town. So he talked to Mark Carone, who was still working closely with me at that time, and was very interested. Subsequently, he made a second visit to look at Duke again. Again, I was out of town. And anyway, he decided to come to Duke anyway and work with me, even though we had never met. I can't remember, but I'm sure I must have talked to him on the phone or something. But I never met him before he came to the lab. So I think all these stories attest to my remarkable recruiting ability and my ability to ask just the kind of probing questions that bring the very best people to my laboratory.
That's hilarious. I can add two more stories to that. So I had a podcast episode with Dr. Arun Shukla, who said that he wrote you a letter and you said, you know what, just come on, let's chat. Yeah. And Michel Bouvier, who I recorded a podcast episode with him last week, and he said that he called you up and you told him you're lucky because I usually don't pick up the phone when I don't know who's calling.
That's true, absolutely. And so all this comes back to fate. I'm kind of mystical in that sense. I sort of believe, well, what's supposed to be will be, and if the right people come, and we just sort of mesh. Bouvier, of course, was here during just an unbelievable golden era in my laboratory. Yep. All of the following people, and I hate to do this because I'm going to miss somebody, but all of the following people at least overlapped in time in my lab in the 80s. Okay, so you had Rick Sirione, you had Martin Losey, you had Brian Kobilka, you had Henrik Dolman, you had Jeff Benovic, you had Michel Bouvier, you had Steve Liggett, all within a 5-year period. And there were others, and I apologize. I'm probably missing some very, very important ones. But just in this 5-year period, there was just amazing concentration of people. But of course, none of them were who we know today, right? I mean, yeah. It was Jeff Benovic before he was Jeff Benovic, Brian Kobilka before he was Brian Kobilka, Bouvier before he was Bouvier. Just like the group I told you about of Yellow Berets at the NIH. I mean, it was Joe Goldstein before he was Joe Goldstein, Tony Fauci before he was Tony Fauci. I think the important thing to take away, or an important thing to take away, especially for the young people just starting out there, is, we all started just like you. I mean, Bob Lefkowitz wasn't always Bob Lefkowitz. I mean, I don't know who he was, but anyway, certainly not anybody that you would look up to or want to emulate.
Well, you got to a point where you learned how to deal with failure, and you also asked the right questions, and you were interested in topics that motivated you, to go and do the research. I was talking to Brian Roth as well. I don't know if you got a chance to listen to that episode, but Brian was repeatedly told that he would never make it as a basic scientist.
Who told him that, I wonder?
I don't know, but apparently that person apologized to him over the years. But he was interested in a specific set of questions that led him where he is today. And I'm pretty sure it's exactly what happened with you and with all of the people who overlapped, who were in your lab, because you were asking questions that you were interested in answering.
Well, let me, you hit on a point that I think is worthy of emphasis. And it relates to mentoring and also to another interesting phenomenon, which is of interest to me, of lineages in science. So you said that I had worked on things that were interesting and important. And of course, one of the most, if not the most important thing in any scientist's career are the decisions he or she makes about what are you going to work on, right? I mean, in the moment you decide what your question is, you're pretty much setting the path for how important that work is going to be. In other words, if you choose an unimportant or trivial problem, you may solve that problem up, down, and sideways, but it doesn't matter and nobody cares because it's a trivial, unimportant problem. On the other hand, you might choose a really, really important problem, but it's beyond you. And so you spend years working on something which, if you could have done it, would have been great, but you can't do it. And you're fated not to be able to do it. So there are really two antithetical ways you can fail in research. I call them high output failure and low output failure, which the clinicians in the group will recognize as terms we use in talking about different forms of heart failure. So high output failure is you choose to work on trivial problems, you solve them one after the other, you publish dozens, if not hundreds of papers, but nobody cares, nobody cites them. And despite having 500 papers on your CV, you're an abject failure. At the other extreme, low output failure means you really have an eye for important problems, but you don't have a good sense of what's tractable and what's not. So you spend years working on important things that you never solve, and you never publish much, and so that's low output failure. So the question is, how do you develop the ability to choose on the spectrum from trivial to really important? What you want to do is you want to go as far as you can in the direction of really important problems, but without going over that cliff of it's important, but you can't figure it out, okay? Well, that's obviously a very subtle judgment call, and not anything that anybody can tell you how to do. So how do you learn how to do that? Mentoring. And by mentoring, I don't mean that the mentor explains it to you, because the mentor can't. You just got to watch. And that's what mentoring is all about. You essentially apprentice yourself to a scientist, and you live with them for a period of years, and you watch. Now, of course, to watch, they have to make themselves available and transparent. If they don't, all bets are off. And from this mentoring experience, day in and day out over years, you absorb, I believe, the values of the mentor. How does he or she choose a problem? What does he seem to look for? What is he or she drawn to? When does he stick with something, even though it seems, when does he keep hammering away? You know, or when does he drop it? The old business of, got to know when to hold them and when to fold them. Yep. When does the mentor keep driving towards a problem and ignore potentially interesting side issues which come up? And when does he stop and say, that's kind of interesting and surprising. We better pursue that, even though it's a bit of a diversion. All these things, nobody can tell you how to do any of that, okay? So the key thing of being able to choose interesting problems that are nonetheless tractable is so important. And it's a talent based on a set of subtle values that I believe you acquire from your mentor. Now, in support of this idea that there are all kinds of transferable elements to doing really important science, to attest to the fact that there must be such transferable elements, I cite the existence of scientific lineages, which you see everywhere. Yep. And now you were just speaking about some of the folks who trained in my laboratory, and that's just a fraction of the ones that you know.
Yep.
In that essay I mentioned before, Serendipitous Scientist, I have, there are only two figures, but one figure two is the scientific lineage, scientific family tree of, it was then 9 of us, Yellow Berets from the 60s, who went on to win the Nobel Prize. And I traced the lineages of each of these people back through about 5 generations. Now, what you find is that of the 9 of us, 4 trained directly with a Nobel laureate.
Wow.
The other 5, that includes me, didn't train with a laureate, but you only have to go 1 generation back. All 5 of us have a Nobel laureate as a scientific grandparent. Okay? Some have 2. And then back. Now, so if you take these, I don't know, maybe 50, 60 people on this scientific chart, about half of them are Nobel laureates. Okay, so starting with these 9 Nobel laureates here, you have all these other Nobel laureates. And I submit that this is just one extreme example of scientific lineages. And as I say, it's all a matter of developing a certain sense of taste, a certain style. Yeah, I got to tell you, nothing gives me more pride when I read papers from my alumni. And, you know, I look at it and I say, that's nice. Yeah, good problem. Yeah, you've done good. So anyway, that's my view on sort of mentorship and choosing important scientific problems.
What kind of a mentor were you? And how did you evolve over the years?
Well, that's a very interesting question. So in my dotage, in which I find myself now, I think a lot about such things as mentoring. But certainly for the early years of my career, and I would say for much of my career, I did not think about mentoring at all. I realize, and in fact, I would cite in that context a comment about one of my favorite philosophers, a guy named Yogi Berra. Yogi Berra was the catcher for the New York Yankees, a Hall of Fame player that I idolized growing up in the 1950s. And all sorts of quotes are attributed to him, probably mostly, erroneously. But one thing he did say was he was being interviewed one time about the fact that he was one of the greatest clutch hitters in the history of baseball. He had a very high batting average, like about 290 lifetime. But if you confine this to clutch situations, it was like almost 400. Bottom of the 9th inning, team's down by 1, Yogi at the plate, you know he's going to drive in the winning run. So he was asking, Yogi, they say, how do you do it? When you're up there in the clutch, in the bottom of the 9th, the game is on the line, what are you thinking about? And he responded, think? How are you going to think and hit at the same time? Okay, now, that was my approach to mentoring. I was too busy doing it to be thinking about what I was doing, okay? So I think it came naturally to me. I'm a naturally gregarious person, and to me, science was a shared activity. So I had this tremendous enthusiasm and focus on the research, and my attitude was, come along with me. Let's share this together. Let's do this. Let's just immerse ourselves. And of course, it never occurred to me that I was mentoring, but of course, that's what I was doing. I was sharing with them everything about how do I do it, because it was all together. Now, is that the only way to mentor? Absolutely not. And if I had to, and I have been in the position on many occasions in recent years to participate in panels and discussions about mentoring. And when people ask me, well, what's the secret to mentoring? I say, well, there's no one secret, but one thing for sure is true. You got to be true to yourself, true to your authentic self. I mean, for example, you couldn't find 2 more opposite personalities than me and Brian Kobilka. But I think we both have reputations for being excellent mentors. So that tells you immediately that there's no one right way to do it. I mean, he's an introvert, I'm an extrovert. We obviously do things very differently. But for each of us, our own style seems to work. So I think one of the keys to mentoring is to do it your own way. But I think you need to be sensitive to the fact that it's a huge responsibility.
It is. And if I might add, I think there is the importance of the mentor and the mentor's styles, but I think the mentees also need to find the mentors with whom they can learn.
Absolutely. And let's face it, not every two people can mesh in just the right way.
Exactly.
I find it delightful that, many of my people who've come through my lab, I love these folks. It's like a family. If possible, even more rewarding, I would have to say at this point in my career, than the scientific knowledge we put forward, is for me just knowing all these folks are out there. And you can imagine how many generations there are. I mean, the generation time in science is fairly short. I don't know whether it's 5 years, 7 years, whatever. But I don't know how many great grandchildren I might have. It's been just very, very exciting aspect for me.
I have one other question. We didn't talk too much about science. I think everyone in the field has read your papers, has seen some of your talks. I've seen at least 2 or 3 of your talks when you went through your 25, 30-year career. But I asked this to all my guests. Do you have a favorite GPCR?
Well, for me, of course, it would have to be the beta-2 adrenergic receptor. That is the receptor that I worked on pretty much exclusively for about the first half of my career, which was 25 years.
Yeah.
And with that receptor, we basically, used it to, we developed a radioligand, we purified it to homogeneity, and we cloned it, a first for each of these, which led to our discovery of the GPCR family. It was using the beta receptor that we discovered a novel enzyme which phosphorylated it, which we at the time called BARK, for beta adrenergic receptor kinase, today GRK2, which led to our discovery of the GRK family. And using the beta receptor, we discovered the beta arrestins. If you take those findings, those are sort of really the central findings, in terms of, when you think about it, there are really only 3, there are really 4, the foremost, central components of these signaling systems are the receptors themselves, the G proteins, the GRKs, and the berestins. And 3 of those 4, everything other than the G proteins, we discovered using the beta-2 adrenergic receptor. And of course, Gilman's work discovering the G proteins, which was contemporary with a lot of our work in the 80s on the receptors, was also done largely with the beta-2 adrenergic receptor. So I would argue that the 2 most important receptors, GPCRs, from the point of view of what we know about them today, were rhodopsin and the beta-2. And of course, it was in our original cloning paper in 1986 of the beta-2 receptor where we first perceived the homology and analogy with rhodopsin. So that was the key breakthrough that told us, oh my God, they're all going to look like this.
How did you feel when you discovered that, oh my God, they are all going to look the same?
That was probably the closest I've come in my career. I've had a number of what people like to describe casually as eureka moments, but that was probably the biggest eureka, because inconceivable as it seems today, we had no idea before we cloned the beta receptor that it had any structural relationship with rhodopsin, or for that matter, with any other G protein coupled receptor. And in fact, when we were working on the cloning in the mid-'80s. When we finally, and again, there are all kinds of stories in the book about all of this stuff. I mean, every one of the discoveries I just glossed over was associated with innumerable stories of failure and competition and all sorts of things. But when we were working on the cloning, when we finally, after several years of just abject failure, when we finally had the right clones, and we're doing the sequencing, and it wasn't like today where the sequencing would have been done in one day. It took a number of weeks, and as word got out on campus, even though we were trying to keep the secret because we were involved in a big competitive race, as word got out among my closest colleagues and neighbors, scientific neighbors on the campus, that we had clones for the beta receptor and were sequencing. And they would come to me and they say, "What does it look like? What does it look like?" You know, because it was already in the days when people were finding gene families, etc. And I said, "So far, it doesn't look like anything." I said, "What?" And that's what we expect. It's the first member of the family, if there is a family, and it doesn't look like anything. It was only toward the end when we had most of the sequence that we could perceive the relationship to rhodopsin. And it was a total shocker. I've gone back to the literature on several occasions to try to find out, did anybody predict in advance that there would be a relationship structurally between rhodopsin and the beta receptor? Because think about it. We knew at the time already that rhodopsin, like the beta receptor, was coupled to a G protein, transducin in that case.
Yeah.
And that the transducin was then coupled to an enzyme, a phosphodiesterase, which regulated the levels of a cyclic nucleotide, cyclic GMP. So functionally, already it was clear that there were functional analogies between rhodopsin and the beta receptor. But nobody, ourselves included, was smart enough to make that prediction.
Who were you competing against?
So for the cloning, we were competing against a team. So Elliot Ross, who had been a postdoc with Gilman and was now an assistant professor, both of them by that time at Dallas Southwestern. We had been competing with Elliot and Al on the reconstitution studies. We pioneered all the techniques for purifying the receptors, which involved creating the radioligands, developing the solubilization procedures, which in those days used digitonin, but it was a very crude material. And we developed the affinity chromatography system. So Elliot Ross, a fine biochemist, had appropriated all of those published techniques. And while we were working on the beta-2 adrenergic receptor, he began working on the Turkey beta-1 adrenergic receptor. And he was able, to his credit, to use all of our techniques to purify that, including our affinity chromatography and everything else. And then he sent protein to a scientist named Axel Ulrich, who was a staff scientist at Genentech. Now, Genentech at that time were the world leaders in this newly developed cloning technology, which was brand new. I mean, we were all biochemists and none of us knew anything about this. Genentech had amazing resources, and they used microsequencing techniques on the Elliot sample of Turkey beta-1, got several stretches of amino acid sequence, used that to design probes, and set out to clone it. We were aware of this competitive effort. Granted, it was on the beta-1 and we were studying the beta-2, but we didn't know anything about cloning. And Axel Ulrich was probably the premier guy with the technology in the world. He had cloned I'm trying to remember, he cloned a number of the growth factor receptors, perhaps starting with EGF and others. And he was just legendary in the field. And we figured this is tough. So we teamed up with some people from Merck through Kathy Strader who had been a postdoc in my lab. And they were helping us with the molecular biology, but we were getting nowhere. And then rumors started to circulate, worse yet, that they had clones at Genentech, and we still didn't. And it was driving me absolutely crazy. I mean, I'm not going to deny for a moment that I am inherently a competitive individual. You know, I'm mellowed out now, but certainly back in those days, in my 30s and early 40s, I was very competitive. And I guess I had come to believe at some level that successfully cloning the receptor and figuring out what its sequence was was sort of my birthright at this point. We had developed every technique that led up to this. And here was this other group that took all our techniques, which entitled to do, we published all of them, and now they were beating us. And it was driving me absolutely nuts. And the guy leading the effort in my lab was Brian Kobilko, of course. And prior to that, I had him working on some other cockamamie projects, which went nowhere. And at some point, after failing for a while on that, he came to me and he said, look, he says, I would love to learn this molecular biology stuff. Why don't you let me in on the cloning project?
Yeah.
I said, what, are you suicidal? I said, you're already not getting anywhere with these other projects. This is going nowhere. We're coming from behind. You sure you want to do this? He said, yes. So I sent him up to Merck, where he spent a few weeks learning the rudiments of recombinant DNA technology. And he came back and set everything up back in my lab. And so then we learned that we're behind. And I remember sitting down with Brian one day and saying, you know, we got to do something different. We've been screening. At that time, most of the, this is before PCR, okay, you know, we've been screening these plasmid libraries for a year now, and we're getting nowhere. People were just beginning to develop phage libraries, okay? Plasmid libraries typically had, in a good one, 200,000 or 300,000 independent recombinants, whereas the newer the newer phage-based libraries had as many as 5 million, 8 million clones. Now, little did we know that at that time, that the mRNAs for these receptors were so rare that typically their abundance might be 1 in a million. Okay. Wow. So in screening plasmid libraries with 200 or 300 independent recombinants, you were doomed. There was no way you were going to find what you were looking for. We didn't know any of that. And I said to Brian, you know, come up with a list, if you can, in the next few days, of other things we could do differently. I said, I don't know what, but just anything. I said, because we're going to lose this race if we don't pull a rabbit out of a hat. I remember a day or two later he came, and I somewhere in my files I still have the piece of yellow legal paper that he or I wrote things down. I think he had 5 or 6 ideas. Not the first idea on that list, but somewhere on that list was try screening a genomic library.
Okay.
So, okay, that's different. So called up the guys at Merck, who were the molecular biology experts, and said, what do you think about making a genomic library and screening that? They said, that's a stupid idea. I said, what did you get from that? I said, well, we thought maybe we'd get a better probe. We were using it, from our purified beta receptor, we had gotten a total of 5 stretches of sequence by microsequencing, totaling about 80 residues. I think the longest one was 19 or 20 residues long. That was the basis for our probes. And they had been screening libraries at Merck. We were screening them at Duke, and we were getting nowhere. So we said, what do you think about a genomic library? They said, it's a stupid idea. We said, why? They said, well, because, the gene is going to have all kinds of introns, so you'll get, at best, a little exon. What good is that going to do? To get the genomic structure, you'll be nowhere. So Brian and I talked about it, and we didn't really have a compelling argument. We said, well, the hell with them. We're going to do it. Nothing else is working. So Brian made a genomic library, and we sent it up to them anyway. We said, why don't we both screen it? The very first screen, boom, both labs pulled out beautiful clones. Within a week, we could tell from the restriction mapping that they all overlapped. And so both labs set to sequencing. And of course, as I said, it took a number of weeks. Yep. But then the strange thing happened. The reading frame, which was an open reading frame, stayed open. And as we kept adding, it stayed open. We kept waiting for the intron. No introns. Make a long story short, there were no introns. The gene for the beta-2 adrenergic receptor turned out to be intronless. It was only the third mammalian gene to that point ever found without introns. You want to talk about fate. And so, in one fell swoop, it was done.
Wow.
So now you see why I believe in fate and serendipity, and whatever is meant to be is going to be. And so, in no time at all, we were done. We published the paper on May 1st, 1986 in Nature. Meanwhile, as luck would have it, the folks at Genentech ran into trouble. They had about half or 2/3 of the sequence of the beta-1 adrenergic receptor, but they could not find clones that encoded the most amino terminal part. And there they were stuck for months. Wow. And so we finished well ahead. And that's the story of the cloning. It was very competitive. And it was just it was a real high when we got there.
How did you celebrate that?
Well, it's interesting. I sent the team, without me, I sent the team out to lunch, and I rented them a limousine. Okay, there were about half a dozen people, including Brian and his wife Tang Sun, who also worked for me, but specifically assigned to Brian, Tang Sun. So about 6 of them, including Henrik Dolman. And I sent them out to lunch at a restaurant of their choice. And in the book, I have a picture of them with the limousine. And it shows the restaurant they chose, which was it's either a Hardee's or a McDonald's. And they went through the drive-through lane in this big, long, rented Cadillac limousine. So more details of that story are also in the book.
That is phenomenal. So we've talked about your eureka moments and all the contributions that your work on the beta-2 adrenergic receptor brought to the field. And we talked about this right before we hit the recording button. Everyone, the way you're perceived as this guy who works hard and gets things done and, gets these discoveries out. Can you tell us a little bit more about the times when things didn't work out and how did you manage those?
Yeah. So my first experience of failure in research, as I mentioned, came at the NIH.
Yeah.
And I was really miserable because I had not yet realized that research is mostly failure. I remember I was having lunch one day with not one of my mentors, but one of the senior scientists down the hall. And this was probably about 6 or 8 months in. And he knew how miserable I was. And I would talk to him about it. He said, Bob, let me tell you something. He says, do you have any idea what the difference is between an average scientist and a world-class scientist? I said, no. He says, well, for the average scientist, maybe 1% of everything they do might work. He says, but for the world beater, for the potential Nobel laureate, he says, it might be as high as 2%. And I thought about that. So this guy's telling me that even for the best of the best, 98% of everything they try is failing. And I have passed that along to many of my trainees. And, there's no easy way. Some people are constitutionally better equipped to deal with failure. But I see one of my most important functions in the lab, is sort of keeping people afloat emotionally, even as my mentors did when things were not going well. The key is to be continuously appraising whether your strategy is a sound one, to reevaluate. But if you think it's sound, then you stick with it. And so I think one of the most important mentoring functions is helping people to learn about dealing with failure.
I could not agree more. You had mentioned that every year at Duke you give this seminar on how scientists can deal with failure and how they can, overcome these situations. Any, top 3 advice that you would have for the young scientists listening to the podcast?
I don't know if there are specific things I could say other than you've got to fight not to lose confidence in yourself. And you just need to realize just how hard it is. I mean, if all you wanted was to succeed in getting your experiments to work, that's not hard. I mean, I could give every graduate student and every fellow who comes to my lab right off the bat projects that are bound to work. But if you, and for some people that's, that doesn't work for me in my lab. But for people who really are dedicated to wanting to do important work, you have to accept that the price of that is mostly failure. In fact, I'll tell you another story. So there's a faculty member. He's not young anymore. I've mentored this. He didn't train with me, but he has collaborated with me a lot. He's at Duke now. And so I had an interesting experience with him, which apparently had a very marked effect on his career, or so he has told me. So it was the week between Christmas and New Year's, quite a few years ago. And I was sitting in my office doing something that I have done basically since the beginning of my career. I don't know why or how I started this, but during that week, I sit by myself for a number of hours and I write down everything that I want to work on in the year ahead. It's sort of a year plan. Mm-hmm. Needless to say, it gets completely changed in the course of the year. And I take those things and then I prioritize them and I put them into bins in terms of what I think their priority will be. I think, I have a priority 1, 2, and 3. And then for the most part, I only work on priority 1 things. Okay. Then a year later, during the same week, I pull out the piece of paper and I go through everything that's on it. And I make a note as to what happened. The possibilities are stupid idea, decided not to pursue it. Stupid idea, wasted 6 months on it. Possibly good, we've been working on it for 8 months now and no progress, but I think this is going to work. Or we did it, paper in progress. And I keep those and I look at them. Anyway, so he came in when I was doing that exercise. He said, "How's it going?" I said, "Fine." He said, "What are you doing?" I explained. He said, "Oh, no kidding." I said, "Well, how are you doing?" I said, "What kind of year have you had?" He said, "Oh, I've had a great year." I said, "Tell me about it." So we went on to recount. He said, "Almost everything I'm working on is succeeding. It's amazing." He says, "It's said like what percent? Close to 100%. Everything that we're doing is working." And then he said, "How about you? What percentage of the things you're working on this past year are working?" I said, "At best, maybe 20, probably lower." He said, "What are you talking about?" I said, "Oh, that's good." He said, "What do you mean that's good?" I said, "Well, if I get anywhere near 50%, I'm unhappy." He says, "Why?" I said, "Because if half of everything I'm trying to do is working, I'm not taking on very challenging stuff." Exactly. So, I really stick around 10 or 20%. Well, he was shocked. But I can't tell you how many times over the intervening years he has mentioned to me how he took that to heart. And he's actually done some really important things since. So it just makes or emphasizes the point that if you want to do important things, you have to be prepared to fail. And I know our lab's been kind of prolific over the years, and people say, oh my God, everything you touch turns to gold. What you're seeing is a trivial fraction. Most of what we do fails, okay? You have no idea. But that's science, absolutely.
It reminds me of this picture I've seen online when you see just the tip of the iceberg, and under it, 90% of the iceberg is actually underwater and you cannot see it.
Absolutely true. Absolutely true. And, people only see, what you publish. What they don't see is what you don't publish, because it's either negative or it's confirmatory or whatever.
Or it's not novel. Before I let you go, tell us about the Nobel Prize. How we've heard about it, we've read about it, but how did you feel when the phone rang?
Well, so that's an interesting question. And you recall, if folks want to read more about it, the book is called The Funny Thing Happened on the Way to Stockholm, and I've got a whole couple of chapters on that experience. Suffice it to say, the Nobel Week experience, and it's actually more like 10 days when you're in Stockholm, is much the most remarkable experience I've ever had in my life. It begins with a call during that Nobel Week that we all know about. I had an interesting relationship with the Nobel Prize in that people had been telling me for years, or predicting for years, that I would win the prize. And they would say, when do you think you're going to win the prize? Well, why haven't you won the prize already? And so, understandably, for a while, when it would come around that Nobel Week announcements, I wouldn't say I was sleeping by the phone, but, I was aware of it. But years came and went, and nothing happened. And then in 1994, Rodbell and Gilman won the prize for discovering G proteins. And a lot of people said to me, well, they could have included you for receptors and G proteins, et cetera. So much of the work was interrelated, but it didn't happen. And I was a little disappointed, but not as much so as I was 10 years later in 2004, when they gave the prize to Buck and Axel for discovering the olfactory receptors, which they did in 1991, 5 years after we published on the beta receptor. And in fact, the key to their success was they came up with a very creative degenerate PCR strategy, where they took something like, I don't remember the exact numbers, let's say 8 or 9, which was the number at that time, 8 or 9 of the published sequences of clone GPCRs. And based on that, they developed this degenerate PCR strategy and discovered the olfactory receptor family. And of those 8 or 9 sequences, 6 had been published from my lab. So basically, absent our body of work, that doesn't happen. And still, no, we weren't included. So that bugged me even more. But okay, obviously one is not in the business to win a Nobel Prize, thank goodness, because it didn't seem to happen. And so I finally got past that. And I must say, by 2012, I didn't really think about it very much, except during Nobel Week, I was interested. I mean, I pretty much assumed that for whatever reason, I was not going to get that prize. And that was fine. I was at peace with that. And Monday came and went, which is the day that the medicine prize is announced. And a big surprise. I mean, I wasn't included. Shinya Yamanaka won for iPS cells. Then at 5 a.m. on Wednesday morning, the phone rings. My wife answers it. And she hands me the phone and she says, "It's for you. It's from Stockholm." And so at that moment, I realized what was going on, but was already confused. And sure enough, a woman with a delightful Swedish accent says, "Professor Lefkowitz, I'm going to give you over to Dr. Sven Leiden, the chairman of the Nobel Prize Committee in Chemistry. He has some good news for you." And so by then, of course, I realized what was going on. He tells me. And I have to tell you, my first reaction was not like jumping up and down and whoop-de-doo, it was more a sense of relief, I think, and quiet satisfaction. The monkey's off my back. I realized in that moment that I would never again have to answer the question, Bob, when are you going to win the Nobel Prize? And I didn't. So that was the start of it. I asked, well, am I sharing this with anybody? Because there were certainly a bunch of possibilities.
Yeah.
And they told me that I would be sharing it with Brian Kobilka. And of course, that brought a tear to my eye to know that it was one of my trainees that I was going to share it with. I said, can I call him? They said, give us 15 minutes. We want to tell him first. And so I did just that. And I went inside and made a pot of coffee for me and my wife. And that is a surreal time, that next hour, because it's all dark. And they tell you can't tell anybody about it until the announcement is made. So you know that you're the only people in the world who know this. It's going to be big news. I have 5 children and grandchildren and innumerable colleagues and people I wanted to share it with. But for the next hour, all we could do was sit and drink coffee, which I did in abundance. The only call I made was to Brian, and we chatted about it and congratulated each other. And then the next 2 months after that are just a blur. There's so much to do in the 2 months before you actually make the trip to Stockholm. And I mean, highlights for me during that period were I was honored at Center Court in Durham at a Duke basketball game. Coach K and the team presented me with a jersey with the number 1 on it, my name. And I was standing at Center Court, and the Duke students who sit closest to the floor were just going crazy. This was about a week after the announcement in October. The coach had called me up and said, why don't you come down? We're going to honor you in a ceremony. And so the students were all there, and I could hear them, I could see them pointing at me. So I'm standing Center Court. They've just put the jersey on me, and the students are all looking at me and chanting something and pointing like this. And at first I couldn't get it. And then it came into focus. They were chanting, he's so smart. He's so smart. So that was pretty exciting. And then the other thing was that it's been a long-standing tradition, not surprisingly broken by President Trump, for the President of the United States to invite all American Nobel laureates to his office to be congratulated personally by the President on your way to Stockholm. So a couple of days before we flew to Stockholm, there were 5 of us, including Brian and myself, and our spouses, all went to the White House, and we met with President Obama in the Oval Office, and that was just great. I mean, he was wonderful and very self-deprecating. You remember he, in his first year in office, had won a Nobel Peace Prize.
Yes.
Which many people, I suspect, including him, felt he hadn't quite earned yet. But he did win it. So when we were in the Oval Office with him, he said, what were his exact words? Something along the lines of, he says, I really feel a kinship with you folks, because as you know, I too am a Nobel laureate. He says, but there's a big difference between my Nobel Prize and yours. And he paused and he said, you earned yours. So there was a bit of self-deprecating humor there. And then the 10 days in Stockholm are beyond anything. I've got a whole chapter in the book about what that experience is like. It's just amazing. It's so different, their society, than ours, in that they revere science and intellectual achievement. Whereas here, we revere sports achievement.
Yeah.
Science is hardly something to be revered, but throughout Sweden, they idolize their Nobel Prize winners. And so the whole time we were there, the press were after us. Our pictures were on the front page of the newspaper every day.
Oh.
We were on TV all the time. And you're recognized in the street. You're signing autographs wherever you go. I mean, it's just the most amazing thing. And then you get to the big day, which is always December 10th, the anniversary of
Nobel's death.
Yeah, when there's the ceremony at the concert hall, and then you go to another venue for the Nobel banquet, which is about 1,500 people, and that occurs directly afterwards, or maybe an hour later, and that's 1,500 people. I mean, the pomp and ceremony are beyond anything you've ever seen. The streets are lined. It's a holiday. The streets are lined with people. As each of us has our own limousine for the whole time you're there, and a driver and a guide. I mean, it's amazing. But it's a holiday, and the tradition is for people to hold their own Nobel banquets in their homes. And so the closest analogy I would have for the whole thing would be Super Bowl weekend. So people have Super Bowl parties. So what they do there is the tradition, and the big day, it's all white tie, not black tie, white tie, long coats, and the whole business. So people have banquets in their homes, and they invite friends and relatives. They dress up, many of them, in formal attire, and they have a banquet while watching the ceremony and the banquet on TV. So, I mean, it's covered. It's probably a good 5-6 hours of TV from the start of the ceremony to the end of the banquet. And the banquet's got all kinds of entertainment and people describing what's going on. And so they have their banquets even as the real banquet is going on. So it's, needless to say, pretty incredible.
It's interesting. So you were supposed to give a talk. I was a postdoc back then at Rockefeller University, and you were supposed to come to give the Friday lecture at Rockefeller, I think the week or the day before, or something like that, before the announcement was made. And it took 1 whole year to reschedule your talk.
Actually, I remember that well. No, the problem was I got the call on Wednesday, and I was supposed to leave Thursday morning to fly to Rockefeller to give the lecture on Friday, okay? And it's just not possible. So I canceled that and did it a year later. But I do remember that was the only thing that I had to cancel during the month.
And then the year later, that's when you and I took a picture, which I still have.
Great, wonderful.
I still have. So the other question I had regarding the whole Nobel week is, I've seen this picture where you're in this fabulous room surrounded by a lot of your trainees who went through your lab. Did you have a number of people you could invite, or how does that happen during Nobel week?
Okay, so in terms of the ceremony and the banquet, you get, in addition to your own 2 tickets, you get, I forget, either 12 or 14 tickets. You don't get them, you got to pay for them. They're not cheap, but you just won the Nobel Prize, so you're not worrying about money. But the tickets, I remember, were $500 a piece.
Wow.
But I have 5 children, 6 grandchildren, a couple of very close colleagues, etc. So I really, in the end, I couldn't bring any of my key people with me, or even the president of the university or anything like that. Brian has a much smaller family and actually was able to bring several of his key collaborators with him. Okay. So all of a sudden, but many of the events are open to the public, like the Nobel lectures.
Yeah.
So a movement got started. To this day, I don't know who started it, amongst my trainees and Brian's trainees, to just come to Stockholm for some center cut portion of Nobel Week and just be there together to experience it and to support us. In the end, I don't know, we had 60 or 80 people, I don't remember.
Wow.
Brian and I arranged to have a cocktail reception at the Grand Hotel, which is where the Nobel laureates stay, one day, and they all were there. And that's the picture you've seen, of us surrounded by our trainees and a couple of Brian's collaborators, etc. And many of my key people that you mentioned, trainees, were there. And I mean, I was so touched by that on their own nickel, they would fly halfway around the world in the middle of the winter and hang out, which is just what they did. And then, on the big day, they couldn't come to the ceremony and the banquet, but, they gathered together in various, I guess, bars and watering holes. And together were able to watch the whole thing. And then for the actual Nobel lectures, they actually came to our lectures. So just having them all there was so perfect because, let's face it, science is a team sport. The prize, although it went to me and to Brian, recognized the work of all of the people you mentioned earlier and dozens and dozens and dozens more. So having them all there, and it's interesting, after my Nobel lecture, I was back in the States and I was being interviewed by somebody. And he said, I really appreciated the fact that you mentioned so many of your trainees during the lecture, because what that Nobel lecture was sort of the first time I ever really ran through my whole career's work. And at each point where I showed a key discovery, I mentioned aloud who did the work. And anyway, this guy had listened and watched my Nobel lecture on the website. And he said, do you have any idea how many people you specifically named? I said, no. He said, over 50. So I was really pleased that I had, and an example of just how meaningful that is to people. I have a postdoc, you might recognize his name, but I'm not going to mention it in this context. Had a very successful academic career. And he had lost, at some point along the way, he lost his wife to cancer. And so he had wound up raising two daughters, largely without their mother. Anyway, he came over with all these other postdocs and was at my Nobel lecture with his two daughters. By then, they looked to be late teens. Okay. And I did call him out specifically. Frankly, I didn't know he was in the audience, but he was one of the 50 that I called out. And he was called out for a very significant discovery. And I mentioned his name. Anyway, afterwards, he came up to me with tears in his eyes. And he said, Bob, I can't tell you how much I appreciated your mentioning my name and what it meant to me for my daughters to be here, to hear that, and to know that I contributed to this. And that really made me, pleased that I had done that. But, that's the right thing to do. It's just that you can't mention everybody. That's always the risk.
Yeah.
That's the risk, especially when there's, in my case, over 200 people. So that's the story.
That's phenomenal. So just let me tell you from the other side of the events, we were all ecstatic at learning that you and Brian Kobilka had won the Nobel Prize. A lot of us wished we could go to Stockholm. As I mentioned, I was at Rockefeller. So Tom Sekmar, who was my mentor at Rockefeller, was in Stockholm during the Nobel week. So he came back and told us all these inside stories.
Right.
But I think as you mentioned, I think everyone in the field somehow felt a connection to you and to Brian and to the field and to the Nobel Prize during that week, which is still phenomenal when you think about it.
It's so nice to hear that, and it makes me very happy. I think we all have the sense that the way the Nobel Prizes kind of work is first they decide on a field.
Yes.
That is having a big enough impact and is clearly important enough to, in their view, merit the prize. Then they decide, well, who might we give the prize to? What 3 people or less might we recognize? Yeah. But of course, more than anything, it's a recognition of a field and its importance. And so I think that all of us in the field, GPCR field, they should have a sense of pride. Were it not, sure, I have my contributions to the field and Brian has his, but had the field not matured to the breadth and depth that it had by 2012, there would have been no prize. And, it's interesting, the lag time between the time that future Nobel laureates make their key discoveries and to the time when they win the prize has been gradually lengthening in all of the sciences and currently stands at north of 25 years. That's exactly what it was in my case. So why so long? Well, I think it's because what they are waiting for is for the original discovery, for the import of the original discoveries to become clear by what happened to the field afterwards. And so I think, in that sense, the prize is a prize to the entire field.
I agree. So before I let you go, I have one last question for you. So you mentioned that you have 5 children and 6 grandchildren, and you've won the Nobel Prize. You've contributed with all of these phenomenal discoveries to the field. How did you do it? How did you manage what we call today work-life balance?
Very poorly, very poorly indeed. And I love my kids. I have wonderful relationships with them. It would be a sham to suggest that I could have done this body of work and have been this deeply involved for so many years and been the typical dad going to every little league game and this and that. That said, I did a lot of it, but certainly not as much. And the key, I think, to the success of that was that my first wife, we were divorced many, many years ago, and I'm very happily remarried. But my first wife, Arna, did not have a career outside the home. Okay. So it was a real partnership. She devoted herself to the raising of the children while I devoted myself to making scientific discoveries and being a physician. That gave me a flexibility and a freedom that very few have these days in our line of work, because most scientists that I know are married to other professionals, or frankly, for the young people, almost invariably other scientists, because who do you meet, right? I mean, any of us in a lifetime meets a very tiny number of people, and most of us fall in love and get married, but you're really choosing from a very small group of people, and the people you meet, and the people that you meet are basically the people at work, and in your field. So most families that I know, and for many years, have been 2-career families. And so that involves a lot more juggling and balance than I had to go through, because, I would drop in. I wasn't as workaholic as Tony Fauci, but, Tony didn't marry till very late in the game. I mean, he was probably in his 40s by the time he got married. I mean, there was no time. He was, as you said, having lab meetings at 7 a.m, and he's famous for not going home till about 9 o'clock at night.
Yes.
So this issue of work-life balance, people often ask me what the secret is, and I have to tell them, you're talking to the wrong guy, because I had it easy. I really did.
You had the right partner in that sense.
Yeah, I think so. Yeah.
I think it's a partnership. And I've heard stories about Dr. Fauci, who actually you need to take an appointment with him as a family member to go for breakfast, because he's so dedicated.
That's true. I mean, when you look at what goes on there, I mean, I don't know how he does it all. I just don't know.
I don't either. I think he's just he's just superhuman when it comes to.
He really is. I mean, you think that the guy is about to turn 80. It makes it even more remarkable.
It's he's phenomenal. Well, thank you so much, Bob, for your time. I really enjoyed it. So everyone here listening out there, you can already pre-purchase Bob's book on Amazon, but it will come out on February 2nd, 2021. Please go ahead and purchase it. You'll hear more about all of the stories that we a little bit grazed upon, and obviously the Stockholm week, and as well as all the phenomenal stories that led to that week in Stockholm. So with this, thank you so much, Bob.
My pleasure indeed. I'm glad to have had the opportunity to talk to you today.
Thank you so much. Bye, everyone.
Bye-bye.
Thank you for listening to this very special episode of the Dr. GPCR podcast. I'd like to thank Dr. Bob Lefkowitz, Dr. Randy Hall, Attila Forrest, Shivani Sachdev, and Jin Chung. Don't forget to subscribe to the Dr. GPCR newsletter at drgpcr.com/newsletter. Also, be on the lookout as we will be launching our brand new Dr. GPCR virtual cafe. Email us for more information at hello@drgpcr.com and be on the lookout as you'll be able to register to present your work or your company's technology. Until next time, stay safe and thank you for your time.